Deep inside your DNA, according to reports from July 2026, researchers found evidence of something that shouldn't exist. An ancient human relative with no bones, no teeth, no fossils anywhere on Earth, detectable only because traces of it are still sitting quietly inside the genome of every living person. Before going further, here's an honest correction worth making right at the start, because it matters. Headlines circulating about this story credited the discovery to Grok, Elon Musk's AI chatbot. According to the same reporting, that framing is wrong, and the researchers involved have said so directly. This wasn't a chatbot answering a question. This was computational biology, a genuinely sophisticated statistical method developed by working geneticists, applied to genetic data the way rigorous science actually gets done, then published for peer review. I can't independently verify every detail of this specific 2026 study myself, since it falls outside what I can confirm with confidence, so I'll be upfront about that throughout. But the broader scientific territory this story sits inside, the real, well-established history of ancient human interbreeding, is something I can walk you through with full confidence, because it's some of the most well-documented and genuinely strange science of the last 2 decades. Let's start with what we've actually known for certain since 2010, because it's the foundation everything else in this story builds on. For most of the 20th century, the standard model of human evolution was fairly linear. Homo sapiens emerged in Africa, spread outward, and eventually became the only surviving human species on the planet, encountering other archaic humans along the way, but never mixing with them in any lasting genetic sense. That model held until 2010, when researchers extracted and sequenced DNA from a Neanderthal fossil, and the results were unambiguous. Modern humans and Neanderthals had interbred. If you have European or Asian ancestry, somewhere around 1 to 4% of your genome today comes directly from a Neanderthal ancestor. That same year, and in the years following, an even stranger discovery came out of the same general region. Researchers working in Denisova Cave in Siberia recovered a single finger bone and a molar. Genetic material extracted from remains too fragmentary to even confidently identify by traditional paleontological methods, and sequencing revealed an entirely new archaic human lineage, now known as the Denisovans, distinct from both Neanderthals and modern humans. Denisovan ancestry shows up today primarily in populations across East Asia, Southeast Asia, and especially the Pacific Islands, sometimes reaching several percent of an individual's genome in certain Melanesian populations. Both of these findings, extraordinary as they were, depended on something crucial: actual physical fossils containing preserved ancient DNA. Bones and teeth that had survived tens of thousands of years and could be excavated, cleaned, and sequenced directly in a laboratory. That's a genuinely difficult, but conceptually straightforward kind of science. You find the bone, you extract what DNA survives inside it, you sequence it, and you compare it to modern humans. It's worth appreciating just how difficult that extraction process actually is, because it took decades of dedicated methodological work to make it possible at all. Ancient DNA degrades over time into extremely short, damaged fragments, and any bone that's spent tens of thousands of years in the ground is absolutely saturated with modern contamination, bacteria, fungi, and DNA from every person who's touched it since excavation, all mixed in with whatever tiny scraps of genuinely ancient genetic material survive. Geneticist Svante Pääbo spent much of his career developing the specialized laboratory techniques, ultra-clean extraction protocols, and statistical methods needed to reliably separate real ancient signal from that overwhelming background contamination, work serious enough that it earned him the Nobel Prize in Physiology or Medicine in 2022. Pääbo's own early research, in fact, was repeatedly undermined for years by exactly this contamination problem, with several of his initial published samples later turning out to actually be modern human DNA introduced through careless handling rather than genuine ancient material, a humbling setback that reshaped how the entire field approaches ancient DNA work today, with far stricter clean room protocols now standard practice at any serious ancient DNA laboratory. The genuinely hard problem researchers have wrestled with for years since is this: what do you do when a lineage that once existed and clearly interbred with our ancestors left behind no fossils at all? No bones, no teeth, nothing a paleontologist could ever dig up and put in a sequencer. According to the reports about this 2026 research, that's exactly the challenge a team working on computational genetics set out to solve, not by waiting for a lucky fossil discovery, but by working backward from the DNA already sitting inside living people today. The method described in this research is referred to as TRACE, reportedly developed through work connecting researchers at UC Berkeley with collaborators at Johns Hopkins University and a genomics research initiative called 54Gene. The core idea behind this kind of method, and this part is genuinely well-established computational genetics, not speculative, is that when DNA passes from parent to child, it gets shuffled and recombined at specific, statistically predictable points along each chromosome, a natural process called recombination. Stretches of DNA that share a common, more recent ancestor tend to show less internal recombination between them than stretches with a more distant shared ancestor. By carefully analyzing recombination patterns across a large number of modern human genomes, researchers can reconstruct a kind of genealogical map, identifying which chunks of DNA likely arrived from distant and sometimes previously unknown ancestral populations, and roughly when that genetic material entered the human lineage. According to the reporting on this study, the method was validated by testing it against genetic contributions from Neanderthals and Denisovans, ancestry we already know exists and can independently confirm through actual fossil DNA. The method reportedly identified these known contributions correctly, which is exactly the kind of validation step legitimate computational genetics research is supposed to include before applying a new method to unknown territory. With that validation in place, according to the study's reported findings, researchers turned the method loose on genetic signatures in modern human genomes that couldn't be explained by either Neanderthal or Denisovan ancestry, signatures that had been noticed in earlier genetic research for years, without anyone having a good way to characterize exactly where they came from. What the analysis reportedly found was evidence of not one, but two separate, previously unidentified ancient lineages that had interbred with modern human ancestors, neither of which has ever been linked to any confirmed fossil. The first of these, according to the reported findings, appears to have interbred with modern human ancestors in Africa more than 50,000 years ago, before the major migrations that eventually spread modern humans across Europe, Asia, and beyond. The lineage itself is reported to have diverged from the branch leading to modern humans roughly 800,000 years ago, an enormous span of separate evolutionary history before that eventual reunion and interbreeding event. The genetic contribution is reportedly fairly consistent across modern populations, regardless of ancestry. Roughly 0.5% to 1% of the total genome, which would make sense if the interbreeding happened early, before the population split into the separate migratory branches that eventually became today's global human populations. It's worth pausing on what that percentage actually represents in concrete terms, because a number like 0.5% can sound small until you consider the scale involved. The human genome contains roughly 3 billion base pairs. Even at the lower end of that reported range, you're talking about tens of millions of individual genetic letters spread across your entire genome that trace back to a population with no name, no confirmed physical remains, and 800,000 years of independent evolutionary history separating it from the rest of the human lineage before that reunion occurred. The dating methodology behind claims like these deserves a bit of explanation too, since it's easy to hear a number like 800,000 years ago and wonder how anyone could possibly know that with any real confidence from genetic data alone without a fossil to directly radiocarbon date. The underlying technique relies on something called a molecular clock, based on the well-established, measurable rate at which small mutations accumulate in DNA over successive generations. Scientists have calibrated this mutation rate carefully, using known reference points including comparisons with fossil-dated Neanderthal and Denisovan genomes, which is exactly why validating a new computational method like this against already confirmed archaic ancestry, as the reported UC Berkeley research reportedly did, matters so much for establishing credibility. If a method correctly identifies known Neanderthal and Denisovan genetic segments, and correctly estimates when that ancestry entered the human genome, consistent with independently fossil-dated evidence, researchers gain legitimate confidence that the same method's estimates for previously unknown, undated lineages are similarly reliable, even without a physical fossil to cross-check those specific claims against directly. The second reported lineage is described as showing up specifically in Oceanian populations in smaller quantities and connected to a genuinely fascinating, layered possibility that Denisovans themselves may have interbred with an even older, unidentified archaic population before Denisovans ever encountered modern humans, meaning some of what modern Pacific Island populations carry today could be a kind of inherited genetic echo passed down through more than one archaic interbreeding event stacked on top of each other. Here's where it's worth connecting this reported 2026 research to something I can discuss with much greater confidence, because it fits into a broader pattern that's already independently well documented in published, peer-reviewed genetics research from recent years. In 2023, a research team at the University of Cambridge developed a separate computational method for detecting ancient population splits and reunions hidden in modern genetic data. And, applying that method to large genetic datasets, they found strong evidence that all modern humans descend from a reunion between two populations that had been separated for roughly 1.5 million years, coming back together and interbreeding around 300,000 years ago, with the larger of those two populations contributing roughly 80% of the resulting genetic material, and the smaller population contributing the remaining 20%. That research, unlike the specific July 2026 study I can't independently verify, is genuinely well established and has been discussed extensively in peer-reviewed literature. If the newer reported findings hold up under continued scrutiny, they'd represent an additional, more recent chapter in what's turning out to be a consistent, recurring pattern in human genetic history. Our species doesn't trace back to one single, clean, uninterrupted lineage, but to multiple splits, isolations, and reunions between ancient populations stretched across enormous spans of time, several of which have left no fossil record at all and are detectable only through exactly this kind of careful genetic reconstruction. This pattern also connects directly to a genuinely remarkable confirmed fossil discovery from 2018, when researchers working in Denisova Cave identified the remains of a teenage girl who turned out, through direct genetic analysis, to be a first-generation hybrid with a Neanderthal mother and a Denisovan father. This was the first confirmed physical fossil evidence of an immediate, first-generation offspring between two distinct archaic human lineages. Direct, undeniable proof that these different archaic populations weren't just occasionally crossing paths at the margins of their territories, but were, at least on occasion, forming direct, immediate family relationships with each other. It's a genuinely remarkable single data point that reframes how researchers think about contact between these different lineages, from occasional distant encounters to something considerably more intimate and immediate. Taken together and putting proper weight on which specific claims are well established versus which are more recent and less independently verified by me directly, the honest picture that emerges is this. It's well established, confirmed by direct fossil DNA evidence, that modern humans interbred with both Neanderthals and Denisovans. It's well established, through published peer-reviewed research using entirely different computational methods, that our species carries genetic evidence of an even older split and reunion stretching back over 1 million years, involving a population with no confirmed fossil identity. Whether the specific additional ghost lineages described in the reported July 2026 research hold up under the same level of independent scrutiny and replication is something I genuinely can't confirm myself, but it fits comfortably within a pattern that legitimate, already published genetics research has independently established as real and ongoing. What makes this entire body of research so significant, beyond the specific number of ghost lineages involved, is what it does to the old tidy model of human evolution as a simple branching tree. That old model pictured Homo erectus giving rise to separate, geographically isolated populations, one becoming modern humans in Africa, another becoming Neanderthals in Europe and Western Asia, another becoming Denisovans further east, with occasional marginal contact between them before modern humans eventually outcompeted and replaced the others entirely. What the genetic evidence increasingly supports instead is something closer to a complex, tangled network, populations splitting apart, evolving separately for enormous stretches of time, sometimes for hundreds of thousands or over 1 million years, and then reuniting and interbreeding, sometimes repeatedly, before splitting again or eventually going extinct, leaving their genetic legacy quietly embedded in whichever population survived to carry it forward. This also raises a genuinely interesting question about how meaningful the traditional biological definition of species even is when applied to ancient humans. The classic definition holds that separate species can't produce fertile offspring together, but Neanderthals and modern humans clearly could, since Neanderthal DNA is measurably present in living people today, passed down across countless generations since that original interbreeding. The same holds for Denisovans. If the additional ghost lineages reported in this newer research hold up, the same logic would apply to them as well. At what point does a population that's been separated for hundreds of thousands of years but can still produce fertile, viable offspring with another population stop being meaningfully described as a separate species at all? It's a genuinely difficult question, and one that reflects real ongoing debate within the field of human evolutionary biology rather than a settled, easy answer. It's also worth addressing directly why fossils for these proposed ghost lineages might never turn up at all, since that absence is exactly the kind of thing that invites skepticism, appropriately. The fossil record is heavily shaped by accident and environmental circumstance. Bones preserve far better in some climates and soil conditions than others. A relatively small, geographically limited population, living in a region with poor preservation conditions or simply not yet excavated by modern archaeology, could plausibly leave no fossil trace at all, even after contributing meaningfully to the genetic makeup of every person alive today. The absence of a skeleton doesn't disprove a population's existence. It just means paleontology, which depends on physical luck and preservation conditions, hasn't caught up with what genetics can now detect through an entirely different, and in some ways, more sensitive kind of evidence. This doesn't mean researchers are working entirely without candidates either. Fossil evidence already confirms that several other archaic human species existed in the right general time periods and regions to plausibly correspond to some of these genetically inferred ghost lineages, even without a direct genetic match yet established. Homo erectus, one of the longest surviving human species in the fossil record, persisted in parts of Asia for well over 1 million years, potentially overlapping in time and geography with several of the population splits, and reunions genetic research has identified. Homo heidelbergensis, widely considered a plausible common ancestor to both Neanderthals and Denisovans, is another frequently discussed candidate in exactly this kind of research. The honest current position among researchers working in this space is that connecting a specific genetically inferred ghost lineage to a specific named fossil-confirmed species remains an open, actively pursued question rather than a settled match. And it may well be that as ancient DNA extraction techniques keep improving, some currently unidentified fossil species will eventually be confirmed through direct genetic sequencing to correspond to one of these ghost populations that's currently only detectable indirectly through modern human genomes. There are real, meaningful practical implications tied to this kind of research too, beyond simple historical curiosity. Genetic variants inherited from archaic human populations, whether confirmed lineages like Neanderthals and Denisovans or more recently proposed ghost populations, have been linked in other independently published research to modern health outcomes, including altered risk profiles for certain autoimmune conditions, metabolic traits, and immune system function. Some of these inherited variants appear to have been genuinely advantageous, potentially helping modern humans adapt more quickly to new environments and pathogens as they spread out of Africa and across the globe, essentially inheriting an evolutionary head start from populations that had already spent tens or hundreds of thousands of years adapting to specific regional conditions. Understanding exactly which parts of the genome trace back to which ancient population is a genuinely active, valuable area of ongoing medical and evolutionary research, independent of how many additional ghost lineages ultimately get confirmed. This also has real implications for how we think about genetic diversity across different modern human populations today, and it's worth being careful and precise about what that diversity actually means. Different populations around the world carry different amounts and types of archaic ancestry, reflecting their particular ancestors' specific migration routes, and which archaic populations those ancestors happen to encounter along the way. Populations with deep roots in Pacific Island regions tend to carry more Denisovan-associated ancestry than European populations, for instance, simply as a matter of geographic history and contact, not because any population is more or less evolved or more or less fully human than any other. If additional ghost lineages like the ones described in this newer research turn out to be real and well supported, the expectation would be exactly the same. Variation in which specific ghost ancestry shows up, and in what quantity across different modern populations, purely as a reflection of complex ancient migration and contact history, not evidence of any meaningful hierarchy between populations. Serious researchers in this field have been consistently careful to frame these findings this way, precisely because the history of genetics has, at points, been misused to support exactly the opposite, harmful conclusion. And getting that framing right matters as much as getting the science right. What ties all of this together? The well-established Neanderthal and Denisovan admixture, the independently published Cambridge research on a million-year-old population split and reunion, and the newer, reportedly published findings I can't personally verify in full, is a consistent, increasingly well-supported picture of human origins as something far messier, more interconnected, and more genuinely strange than the clean textbook version most people grew up learning. We are not the product of one continuous, isolated lineage quietly refining itself in Africa before spreading outward alone. We're the product of populations that split apart, evolved separately across scales of time that are almost impossible to intuitively grasp, and then found each other again more than once, leaving genetic fingerprints from encounters and relationships. History has otherwise completely forgotten. If getting the honest, carefully sourced version of stories like this, the real science underneath the sensational headline, is what you're here for, subscribe. Because it turns out the real story sitting inside our own DNA is stranger and considerably better documented than most viral versions of it ever bothered to explain. The bones may never surface, the names of these vanished populations may never be recovered. But their presence, quietly written into the genetic code of every person alive today, is real, measurable, and waiting for exactly the kind of careful, patient, peer-reviewed science that keeps finding new ways to read it.